Usually when we talk about a medical diagnosis, there's this expectation of mechanical precision.
Right, like it's a straightforward physical problem.
Exactly. Like you break your arm, the x-ray shows that jagged white line on the radius bone, and the orthopedic surgeon just points at the film and says, yeah, there's the problem.
The pathology is totally visual. It's immediate. You see the break, you set the bone.
But then you step into the world of genetics, and specifically with chronic metabolic diseases, and suddenly that x-ray machine feels like it's taking pictures through a dense fog.
Yeah, it is definitely not a clean break.
No, it's incredibly murky. So welcome to today's deep dive. We are examining a really groundbreaking June 2026 paper from Nature Genetics.
It's titled Integrative Analyses Elucidate Transcriptional Regulatory Functions of Risk Alleles for Metabolic Liver Disease. Quite a mouthful.
It is. But our mission today is to demystify all that. We want to demystify the dark matter of your DNA. We are going to explore how non-coding genetic variants, the stuff that science used to literally dismiss as evolutionary junk, how they actually act as hidden environment sensing control switches.
Switches that specifically drive MASLD, right?
Right, metabolic dysfunction-associated steatotic liver disease.
Which is currently the most common chronic liver condition in the world. I mean, it affects a massive portion of the global population.
And that leads to a massive paradox for you listening. If you were told that up to 70% of the risk for the most common liver condition on Earth is inherited, you would logically assume we know exactly which broken genes cause it, right?
You'd think so, yeah. We map the genome, find the broken gene, fix the disease.
But genome-wide association studies, or GWAS, they found nearly 100 genetic locations associated with MASLD risk, and almost all of them sit in non-coding regions.
Yeah, the genes themselves, the sequences that actually manufacture the proteins, they're perfectly fine.
Right, they're intact. It's the invisible regulatory switches scattered around them that are altered. So, okay, let's unpack this. What is our DNA actually doing if it's not making proteins?
Well, that paradox is the core mystery this paper tackles. A genome-wide association study is, a statistical powerhouse. You scan the DNA of hundreds of thousands of people and find patterns.
Right.
But GWAS only gives us the zip code of a disease risk. It tells us roughly where a statistical problem is located on a chromosome.
So it's just an area.
Exactly. It does not give us the specific address. It doesn't tell us who the occupant is. And it certainly doesn't tell us what that occupant is doing to cause liver failure.
This is a very blurry map.
Very. And this paper bridges that exact gap. It takes us from identifying a statistical zip code to understanding the precise mechanical single-cell function inside a human liver.
Okay, so to actually read that instruction manual, the researchers used a technique called SNATAC-seq, which is, let's be honest, terrible branding.
Yeah, it stands for single-nucleus assay for transposase-accessible chromatin using sequencing.
Let's break down what that actually means mechanically, because the concept is brilliant once you get it. I like to imagine the human genome as a massive 20,000-book library.
I like that analogy.
Right, because every single cell in your body has the exact same library. A skin cell has the same books as a heart cell.
But SNATAC allows us to see exactly which books are pulled off the shelf and lying open on the desk in a specific cell at a specific time.
Exactly. So how do they actually see those open books? What's the mechanism?
The mechanics are fascinating. They use an engineered enzyme called TN5 transposase. So in our cells, DNA is tightly spooled around proteins just to fit inside the nucleus.
Like thread on a spool.
Exactly. If it's tightly spooled, it's closed off, unreadable. The transposase enzyme acts like a molecular probe. It can only physically insert its sequencing tags into the regions of the DNA that are unspooled or open.
Ah, so it physically can't get into the tightly wound junk.
Right. So by sequencing those tagged regions, the researchers get this high-resolution map of the regulatory elements that are actively in use by that specific cell.
And they did this on a huge scale, right?
Massive. They mapped nearly 70,000 human liver nuclei from 12 individuals. Half with healthy livers, half with severe MASLD.
Okay, so from those 70,000 nuclei, they categorized them into 15 distinct cell clusters. They essentially captured the chromatin fingerprints of every cell type in the entire liver ecosystem.
Yeah, they did. And to make sense of that mountain of data, they use an algorithm called scavenge.
Okay, scavenge. As I understand it, scavenge takes the open books on the desk and cross-references them with those GWA zip codes we mentioned earlier.
Right, and it calculates what they call a trait relevance score.
Right. It's a network-guided approach.
Exactly. Scavenge doesn't just look at single points. It looks at how different accessible regions interact within a single cell type.
Just looking at the whole web.
Yeah, it maps the disease-associated genetic variants down to single-cell resolution. That way, we can see which specific cells are actually using the dangerous mutated regulatory switches.
Okay, I want to pause here and challenge our baseline assumption for a second. When we think of metabolic liver disease or fatty liver disease, we naturally assume it's a disease of the liver cells, right? The hepatocyte.
That's the traditional view, yeah.
Because they are the ones storing the fat. So, logically, the genetic glitches should be happening in the hepatocytes. Does the scavenge algorithm validate that assumption?
Well, it reveals a much more complex picture, actually.
Oh.
Yeah. While they did find that genetic traits related strictly to liver fat accumulation were enriched in hepatocytes, the full progression of MIF and MASLD is a much broader conspiracy.
A conspiracy. Between which cells?
They discovered that the genetic risk variants for the disease are heavily enriched in the open chromatin of non-parenchymal cells.
The supporting cells.
Exactly. Specifically, Kupffer cells, which are the liver's resident immune macrophages, and hepatic stellate cells.
Okay, stellate cells. Those are the architects of the liver's physical scaffolding, correct? They maintain the extracellular matrix.
They do, yes. And under severe stress, they become the primary effectors of liver fibrosis.
Right, they build the scar tissue.
Exactly. They overproduce collagen, creating the scarring that eventually leads to cirrhosis and ultimately liver failure. And the SNAT-exec data was so granular that the researchers identified 3 distinct subclusters of these stellate cells.
Which were S-common, S-normal, and S-MASLD. Right.
Correct. And the SMASLD cells were the ones harboring the highest concentration of the active disease variants.
Meaning they are the ones driving the actual disease progression.
Yes. They exhibited the highest scavenge trait relevant scores by a significant margin, but the researchers went a step further using pseudo-time trajectory analysis.
Pseudo-time trajectory, that sounds like time travel.
It's a computational method that allows us to infer the evolutionary journey of a cell.
Oh, wow. So you can watch it change?
Computationally, yes. They watched as a stellate cell transitioned from a quiet resting state into a highly active, diseased, fibrotic state. And along that timeline, the FSLD risk variants became progressively more accessible and active.
That fundamentally shifts how we view the disease. It means if you are carrying genetic risk for MASLD, it isn't just your main liver cells failing to process fat.
Not at all.
Your immune cells and your structural support cells are genetically pre-programmed to overreact to that fat.
It really highlights why viewing organs as mere collections of identical cells is totally outdated. We have to view them as highly interactive ecosystems.
So true. Okay, time for a quick recap of our first 5 minutes. Let's bullet point this. 1, GSLIs gives us the genetic zip codes, but not the mechanism. 2, SNAT-exec shows us which parts of the DNA are open for business in specific cells. And 3, MASLD is a multi-cell conspiracy involving both hepatocytes and those structural stellate cells.
A perfect summary.
So, question for you listening. Have you ever considered that the background support cells in your organs might actually be the ones driving a disease? It's kind of wild to think about.
It changes everything about how we target therapies.
It really does. Okay, moving on. We have the map. We know the variants are sitting in open chromatin, and we know they are active in stellate cells and hepatocytes. But correlation isn't causation.
Right, just because a door is open doesn't mean a thread is walking through it.
Exactly. Out of the thousands of variants in these regions, how did the researchers prove which specific non-coding typos actually changed the cellular machinery? I mean, they had over 5,300 variants to investigate.
Testing those one by one would take decades.
Right. So what do they do?
They utilized massively parallel reporter assays, or MPRA. This is where the engineering precision really comes into play.
Okay, MPRA. Break that down for us.
They synthesize tiny circular pieces of DNA called plasmids. For every single one of those 5,300 non-coding variants, they created a plasmid with the healthy sequence and a separate plasmid with the mutated disease-associated sequence.
So they have a healthy version and a broken version for every single suspect.
Exactly.
Right.
And to keep track of them all, they attached unique RNA barcodes. Think of them as molecular tracking numbers.
Okay, I follow. They then inserted this massive library of 10,000 plus plasmids directly into living human cell cultures, right? HepG2 cells for hepatocytes and LX2 cells for stellate cells.
Yes. And the brilliant part is, because the plasmids drive the production of RNA based on the regulatory sequence they carry, the researchers can just sequence the RNA later.
Ah, so they just read the barcodes.
Right. If the mutated sequence produces a totally different amount of barcode RNA compared to the healthy sequence, you know definitively that the non-coding typo changes the outcome. It is a functional switch.
That is so elegant.
It is, but they added a crucial layer of complexity. Testing these variants in a peaceful, sterile petri dish doesn't reflect a diseased human liver.
Because genes react to their environment.
Exactly. So they exposed these cell cultures to specific environmental stimuli. They bathed them in palmitic and oleic acid, or PAOA, to mimic extreme dietary fat overload.
Like eating way too much fast food.
Right. And they also applied TGF-beta, which is a cytokine, to simulate a highly inflammatory fibrotic environment.
Here is where it gets really relatable for the listener. It's exactly like a typo in a treasured family recipe.
How so?
Let's say the recipe says add 1 cup of salt instead of 1 teaspoon. If you never actually bake that specific cake, the typo is totally harmless. It just sits in the book.
Right.
But the moment the environment demands you bake it, like hosting a big family dinner, that typo ruins the entire meal. These genetic variants are harmless until the environmental stress of fatty acids or inflammation demands the cell read the recipe.
That is a perfect parallel, and that stress test revealed hundreds of differential activity variants, or DAVs.
DAV, okay.
These are the functional glitches that actively respond to the environment. Let's look at the specific physical evidence. They isolated a variant known as RS6726823.
Okay, let's just call that one the stellate glitch.
Good idea. This stellate glitch showed massive allele-specific activity in the LX2 stellate cells, particularly when stimulated by the inflammatory TGF-beta.
But not in the liver cells.
Nope, it showed almost zero differential activity in the hepG2 hepatocytes. When we look at the actual molecular sequence, this single nucleotide swap disrupts the binding motif for an interferon regulatory factor, or IRF.
Okay, for our listeners, a transcription factor like IRF is essentially a molecular docking ship. It scans the DNA looking for a very specific sequence of letters to land on.
Exactly.
When it lands, it recruits the machinery to read the nearby gene. If the non-coding DNA has a mutation, the landing pad is warped, the ship can't dock, and the gene just gets ignored.
That physical disruption is exactly what happens. By failing to dock, this variant alters the downstream expression of CASP8.
CASP8, what does that do?
It's a gene intricately involved in driving liver fibrosis, so the stellate cell becomes hyperreactive to inflammation because of a single swapped letter in a non-coding landing pad.
Wow. But the researchers also found a hepatocyte glitch, right? Variant RS131500068.
Yes, and this one operates inversely. It acts up strictly within the hepatocyte cells. It sits within a hepatocyte-specific open chromatin region and physically disrupts a binding motif for SREBP2.
Okay, SREB2 is a major player. It's a master regulator of lipid biosynthesis. It's the sensor that detects when cellular cholesterol is low and triggers the creation of more fat.
Exactly. So when this landing pad is warped, the SREBP2 regulatory network misfires. This directly affects a nearby gene called HSD17B13, which controls lipid droplet metabolism.
So the overarching lesson from this MPRA data is that context is everything.
Yes. A non-coding variant only causes disease in the specific cell type it is programmed for under the specific environmental stress it is sensitive to.
Okay, time for another quick mini recap. 1, MPRA tests thousands of variants simultaneously using barcodes. 2, variants act as DAVs only in specific cells. And 3, they require environmental triggers like fatty acids to actually activate. So, listener, think about your own health for a second. How much of your genetic risk is totally harmless right now, just waiting for the wrong environmental trigger to activate it?
It implies that lifestyle interventions aren't just about managing symptoms. They are actively dictating which genetic programs your cells are allowed to run.
That is a wild thought.
Yeah.
Okay, let's follow the data even deeper into the cell. We have the functional switches, but what are they actually turning on and off to cause the fat accumulation?
Right, so the researchers zoomed in on a tightly regulated molecular network surrounding an enzyme called LPL, or lipoprotein lipase.
LPL, what's his job?
LPL is essentially the blood's garbage disposal for triglycerides. Its primary biological job is to clear lipids out of circulation and process them.
Okay, so we want LPL working.
Very much so. And LPL activity is managed by an elegant tug-of-war between two main regulatory genes, APOA5 and ANGPTL3.
Okay, I have a simple mnemonic for this for the listeners. The AAL network, APOA5 activates LPL, stepping on the gas pedal to clear fat.
Yep.
And AGPTL3 arrests LPL, acting as the brake pedal.
I like that. So the MPRA data revealed several key functional variants that tamper with this exact tug-of-war. For instance, variant RS17C1197 sits directly in the promoter region of the ANGPTL3 gene, the brake pedal.
The promoter region being the literal starting line for the gene where the reading machinery attaches.
Exactly. This variant alters the binding motif for a transcription factor called MYC. But the researchers didn't just observe this, they intervened to prove causality.
How so?
They used a technique called CRISPR interference, or CRISPR.
Wait, CRISPR, with an I?
Yeah, interference. Unlike traditional CRISPR, which cuts the DNA strand like scissors, CRISPR uses a deactivated or dead Cas9 protein.
A dead one. How does that help?
They use a guide RNA to drive this bulky, dead Cas9 protein directly to the site of the variant. It parks right on top of the promoter region, creating physical steric hindrance. It physically blocks the transcription factors from landing.
Oh, so they jammed the lock so the key couldn't turn?
Exactly. And the result was immediate. The expression of the ANGPTL3 gene drastically dropped. They ran a similar test on another variant, RS10889356, which sits further upstream in an enhancer region. This one perturbs a binding motif for the GABP transcription factor.
And what happened when they messed with that one?
When they deleted the specific noncoding variant in hepatocytes, the cells suffered massive, visually observable lipid accumulation the moment they were exposed to fatty acids.
So they literally watched human litter cells gorge themselves on fat and failed to clear it just by tweaking one invisible noncoding switch.
Yes. And the complexity doesn't stop with the hepatocytes hoarding fat. They found variant RS1441754, which acts directly near the LPL gene itself, but it only activates in the stellate cells.
The scaffolding cells again.
Right. When this switch is triggered, it upregulates fibrosis-associated genes like COS1A1.
So you have a dual pathway disaster.
Exactly.
One set of noncoding switches forces the hepatocytes to hoard fat and fail at clearing it, while a totally separate switch in the stellate cells reacts to that fat by building massive amounts of scar tissue. It is a coordinated failure.
Driven entirely by non-coding DNA.
Okay, but when the hepatocytes are getting overloaded with these lipid droplets, do they just sit there and accept it? Or is there a compensatory mechanism? Like, does the cell try to defend itself?
Cells are incredibly resilient, and they do attempt to fight back. And this leads to one of the most elegant discoveries in the entire study, the SLC22A3 rescue mechanism.
Rescue mechanism. Love the sound of that.
Right. The researchers identified variant RS474513 sitting in an open chromatin region near the SLC22A3 gene. This variant mutates the binding site for a transcription factor pathway called RUNX.
So the RUNX proteins are trying to dock to mount a defense, but the landing pad is warped.
Precisely. To validate this, the researchers used pharmacological inhibitors. They introduced chemicals that specifically block the RUNX pathway from functioning, artificially mimicking the effect of the genetic mutation.
Okay, and what happened?
As predicted, severe lipid droplets accumulated inside the cells. They proved that a broken RUNX pathway leads to fat storage.
But where does the rescue come in?
Then, they engineered the sick cells to artificially overexpress the downstream SLC22A3 gene.
Oh, so they bypassed the broken switch and manually turned on the defense gene?
And it rescued the cells. The lipids cleared out. This proved definitively that SLC22A3 acts directly downstream of this genetic switch to maintain lipid homeostasis.
That is staggering. Let's do our final mini recap. 1. Non-coding variants control the LPL lipid clearance network. 2, a specific variant controls how the liver handles lipid stress via that SLC22A3 defense. And 3, CRISPR and pharmacological tests actually prove these switches cause the damage.
It's airtight evidence.
So question for you listening. If we now know the exact molecular volume dials causing lipid buildup, could the future of medicine be CRISPR therapies that just manually turn those dials back to normal?
We are definitely transitioning from observing macroscopic organ damage to manipulating the exact microscopic levers that dictate cellular behavior.
Which brings us to the ultimate, so what? We've covered a tremendous amount of molecular biology today.
We really have.
CRISPR interference, chromatin mapping, docking ships. But how does this translate from a petri dish to a patient sitting in a clinic right now? Does knowing the mechanics actually alter clinical outcomes?
This is where the paper separates itself from purely theoretical science and delivers a huge clinical payoff.
Okay, let's hear it.
The researchers didn't stop at molecular validation. They took the 114 specific variants they had functionally proven to be active in their stress tests, and they built a custom polygenic risk score, or PRS.
Okay, a polygenic risk score is essentially a genetic report card. It calculates your cumulative risk for a disease based on the combined weight of multiple variants in your genome.
Exactly. They took this new, highly validated PRS and applied it to real-world clinical data from the VA Million Veteran Program.
Oh, that's one of the largest biobanks in the world.
It is. It contains genetic and clinical data for hundreds of thousands of veterans. They evaluated this PRS against 4 different clinical proxies for MASLD, measuring things like chronically elevated liver enzymes and actual trajectories of fibrosis progression over years.
Let me guess. The custom score blew the old statistical models out of the water.
It completely outperformed them. They proved that scoring patients based exclusively on these 114 proven functional variants significantly improved the prediction of severe MASLD risk. Way more than using standard sweeping GWAS data.
Wow.
Knowing the mechanism fundamentally upgrades predictive medicine. We aren't just looking at a wide statistical zip code anymore. We know the specific addresses, we know which environmental triggers activate them, and we can predict with higher accuracy which patients are going to progress from a benign fatty liver to life-threatening cirrhosis.
That is basically the holy grail of precision medicine. We could look at a patient's genetic profile and confidently say, your non-coding switches are highly sensitive to dietary fat, but resilient to inflammation. So here is your specific intervention plan.
Exactly.
Well, thank you for guiding us through this incredibly dense molecular forest today, unpacking how single-letter changes in invisible DNA cause macroscopic organ failure is no small feat.
It's been a pleasure. It is a profound shift in hepatology and genetics. The more we map these regulatory networks, the more targeted our clinical interventions will become.
And thank you to everyone listening for joining us on this deep dive. But before we go, here's where it gets really interesting, and I want to leave you with a final thought to mull over.
Oh, I like these.
For decades, science arrogantly labeled 98% of our DNA as junk simply because it didn't code for proteins. We assumed it was just, useless packaging material left over from evolution.
We were very wrong.
Very wrong. Because as this deep dive proves, that junk is actually a highly sophisticated environment sensing control panel. It is dynamically spooling and unspooling, constantly reading the room. So if your non-coding DNA is actively adapting to the fatty acids in your diet or the inflammatory signals in your blood, what other environmental signals is your junk DNA quietly listening to right now while you go about your day?
That is a haunting thought.
Right. Is it reacting to your stress levels, your sleep patterns, the air quality? The diagnostic picture isn't murky anymore. We just needed a better lens to see the invisible switches reacting to the world around us. Keep diving deep, and we'll catch you next time.